Hi everyone, and welcome to our video on kidney structure. This is another video in our Module 5 series, and we'll be looking at specification reference 5.1.2c Part 1, the structure, mechanisms of action and functions of the mammalian kidney. So first thing then is that we need to understand a little bit about the overall structure of the kidney itself. Now, one of the diagrams I've given you here on the left hand side, that should hopefully be familiar to us from GCSE, and in reality it hasn't really changed much. So what we've got, the capsule is the bit that surrounds the whole kidney there, and then the outer layer that we can see closest to obviously the outside edge of our kidney is the cortex. The medulla are those dark red triangle shapes in there towards the centre. When we come into this little yellow region here, that is the pelvis of the kidney. And then that feeds down into the little tube going down towards our bladder, which is the ureter. And then we obviously have the blood supply, so the renal artery and the renal vein. And remember, wherever we see the word renal, that just relates to the kidney. So renal artery, bringing the blood into the kidney, renal vein, taking the blood away from the kidney. One of the practicals you may well have done in school is obviously a kidney dissection. And of course, one of those practical assessed tasks that might crop up on your exam papers is actually doing a scientific drawing. So obviously they could give you an image similar to the one I've given you on the right hand side here. And what they could then ask you to do is to draw a scientific diagram of the kidney. Now, obviously make sure you drop back into the practical skills handbook that OCR produced for you here to make sure that you're not making silly errors and therefore throwing away some relatively easy marks on the paper. So sharp pencil is required. Please don't draw in pen, because when you mess it up then there's not much you can do, to be honest. So a sharp pencil. We are not doing the sketchy arty lines when we are doing any kind of scientific drawing. Remember, it's nice, clear, crisp lines. So none of this little feathery line thing going on. Terrible, it's going to cost you a mark. When we're doing the labels, remember we should have a ruler handy, and you are going to draw a ruled line coming off the side of the diagram where you can do your writing, and there are no arrowheads. As soon as you put an arrowhead on, it's costing you a mark again, folks. But as I say, drop back into that practical skills handbook that OCR produced. You can find it on their website, and it takes you through all those rules about how to do proper scientific drawing. Okay, let's jump into the structure of the kidney in a bit more detail then. In terms of the kidney itself, the bit we're really going to be focusing on are those units called the nephrons. And each kidney's got about 1 million nephrons in it. And these are those functional units of the kidney. This is where they're actually going to be doing their thing, so to speak. So the nephrons start in the cortex, that outer region, and then they extend down into the medulla, that inner part. In terms of what happens then, we're going to start with that renal artery bringing the blood to the kidney, remember. So the renal artery is then going to branch off into smaller little arteries called arterioles, and they've got a special name there, the afferent arteriole. Okay. Now each of these afferent arterioles is going to connect onto the glomerulus, which is basically a ball of capillaries. On the other side of the glomerulus, it is then going to leave through the efferent arteriole. And that efferent arteriole is then going to lead to the renal vein, where obviously the blood leaves the kidney. Be careful, it's so easy to mix up your afferents and your efferents here. So we've got a little bit that we can use as a hint on the left hand side there. When we're talking about the afferent, starting with the A, that's the one that is arriving. Okay, so the blood arrives in the kidney through the afferent arteriole. And then it's going to exit with the E through the efferent arteriole. So just make sure you get the afferent as the arrival, the efferent as the exit. When it actually comes to the glomerulus, this is basically what we're looking at. So the blood is going to arrive through the afferent arteriole, and then it flows through those capillaries making up the glomerulus. And you can see it looks like a little tangled ball of string. That little glomerulus sits inside a structure called the Bowman's capsule, which is kind of like a little cup that it's sitting in there, and that then connects onto various tubules in the kidney, which we will look at. Once it's actually passed through the glomerulus, it's then going to leave through the efferent arteriole, which then obviously reconnects onto that renal vein. So, how does this actually work? What structures have we got that relate to the function of the kidney? Well, we need to be able to build up a high pressure for this filtering process to take place first of all. And this is actually achieved through the structure of the afferent and the efferent arterioles. So what we actually find is that afferent arteriole is actually a tiny bit wider than the efferent arteriole. So what that actually means is that because we've got a narrower exit from the glomerulus to obviously the rest of the body than the entry point, what we've got is a little bit of almost a backlog. If you imagine all of these people having like a nice opening to get through first of all, and I'm sure your schools might have this, you're able to get into a corridor quite easily, but then you end up with this narrow bit and it forms almost a bottleneck where people just can't get through because it's a bit narrower. Same kind of idea here with what happens on each side of the glomerulus. So wider at the entry point, so the blood is able to get in nice and easily, but at the exit we make it narrower, so that means we're going to build up the blood pressure. So in the glomerulus we then have this higher pressure, and that's actually going to be able to then force out these small molecules. So things like the water, the glucose, urea, the salts, all of those are going to be forced out of the capillary through those little gaps in the capillary wall and into the Bowman's capsule as the filtrate. Anything that is large, so we're looking at the proteins, we're looking at the actual blood cells, etc., they're going to be too big to fit through the little gaps in the capillary wall, so they stay in the blood. In terms of how the kidney is actually going to do this filtering process, then we need to understand these structures that exist between the actual blood in the capillary and the filtrate in the Bowman's capsule. So we've got three layers that exist between those two regions. First one is the endothelium of the capillary, and we can see that on the diagram in the purpley section. So what we've got are our endothelial cells, and they've got these small little gaps between them called fenestrations. Then we move to our second layer, which is the basement membrane, which is the green band that runs across there. And then finally we come to the epithelial cells of the Bowman's capsule, which is on that yellow section there, and we can see them almost forming like a little chain together. So let's delve a little bit deeper into these individual layers one at a time. So the first one, the endothelium of the capillary then, the bit in the purple. What we've got, as we mentioned, are these gaps between those endothelial cells. And these little gaps are what we refer to as a fenestration. Now, to all intents and purposes, these are like little pores. They're not huge gaps. They're not like massive holes through the wall of the capillary. They're actually relatively small. And what they're able to do is just allow the blood plasma and any substances that are dissolved within the blood plasma to move from the lumen of our capillary and then come into contact with the next bit, which is the basement membrane. Once they get through the little fenestration, they then hit that basement membrane. And the basement membrane itself is made up of collagen and glycoproteins. So notice we've got a little link here back to our biomolecules work in module 2, where we look at the structure of collagen, etc., and the glycoproteins. And those two substances together basically form a mesh, kind of like a net. So what we find here is this little mesh is actually going to act as a barrier to any molecule which has a molecular mass of over 69,000. So anything with that larger molecular mass than 69,000 can't pass through that basement membrane, so it gets stuck. Now, this is obviously going to be quite important to us because that means that none of the blood cells, none of the plasma proteins are going to be able to exit to get through into the Bowman's capsule, assuming the kidney is working properly. So do look out for those questions that then relate to giving you information on things like additional proteins in the urine, etc., because basically that's suggesting that there's an issue at this point here with our basement membrane. Because the membrane, remember, is what's stopping these larger molecules from getting through. Therefore, if larger molecules have got through, that tells us the basement membrane has experienced some form of damage. Third section is our epithelial cells of the Bowman's capsule, and these have a little special name. So these little structures that make up the epithelium is the podocyte. Now, podocytes are basically these little things that almost look like little feet sticking off the cell. So you can see it in the diagram over here. So we've got our podocyte, and then it's got these little protrusions, which are called projections. And we've got those major processes, which are the large ones. And then those major processes have minor processes on them. And the whole idea of these little projections is to basically hold the podocytes a little bit further away from the capillary endothelium than they would be without them. The whole idea of that is it just makes it that little bit easier for fluid to pass into the lumen of the Bowman's capsule. It's just given that little gap for the actual fluid to move between those cells. Now, assuming that we've actually managed to get something through those three layers, then it's going to end up in obviously the lumen of the Bowman's capsule. And from there, it then passes into these tubules. So the first tubule it's going to enter is the proximal convoluted tubule. And if we have a little think about the word proximal, if we think of proximity, if something is in close proximity, that means it's nearby. So proximal convoluted tubule is the one that's closest to the Bowman's capsule. After that, it then flows down this long U-shaped tube, which is the loop of Henle. And then from there, it will enter the distal convoluted tubule. And I always think if you think distal like distant, then that's further away. Okay, so it's the part of the tubule that's further from the Bowman's capsule there. So 3 sections to our tubules: proximal convoluted tubule, loop of Henle, distal convoluted tubule. Now on your exam you can actually get away with PCT and DCT if you want to go with the shorthands for the 2 tubules. When we actually get to that distal convoluted tubule, what we find is that many of those nephrons are going to connect together then. And remember, we've got like 1 million of them in each kidney. So they're all going to connect together to these collecting ducts, which is this little structure here with all those little side branches. These are all going to be different distal convoluted tubules coming in. And then that structure, the collecting duct, is what's going to pass through the medulla into the pelvis, which is this little yellow region here on the diagram, and then obviously the pelvis connects onto the ureter, which goes down to the bladder. Hopefully you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next video is uploaded. There's also additional resources on the website to help you with your A-level biology studies.